this semester, and this was a review session on metabolic biochemistry. Thought I'd share in case it could help anyone. Not nicely formatted or anything like that. Kind of skips around, but if it helps.
So although we can kind of talk about pathways, like that's probably trying to emphasize glycolysis, and like that's probably the TCA. We have so many different ways we can get in and out of these various quote unquote pathways. But we often focus on just kind of like this simplified view of things, where we think of it nicely as like, oh yeah, the sugar comes into a cell and you get broken down to glycolysis, and that goes through the citric acid cycle and oxidative phosphorylation, and voila, you've got energy. But maybe your cells don't need energy. Maybe your cells don't need ATP so much, but they really do need nucleotides because, well, they need to go make some DNA. Or maybe the cells just need to store glucose so that it's not just building up inside. And so there are lots of different things that you can think about happening when that glucose enters the cell. And there are lots of different ways we can get to places like the citric acid cycle. So yes, the citric acid cycle, it often stands out on various charts and things. And one of the reasons why is because there's so many different ways that we can get things into and out of the citric acid cycle. And so it's really like this central hub. And so I want you to kind of think in terms of, not so much in terms of starting with glycolysis all the time, but think in terms of, well, often what we're really talking about, if we want energy, we're thinking about how we get electrons to the electron transport chain. We might not be getting electrons from sugar. We might be getting electrons from fat. We might be getting electrons from protein. But ultimately, these are all, if we want ATP, these are going to end up in our mitochondrial matrix, going through the electron transport chain, passing those electrons from these different complexes to ultimately to power the pumping of protons. The proton concentration builds up, flows through this ATP synthase, turns that, and voila, we get energy. And now, once we kind of have that end in mind, we can think back about how we can get to different beginnings. Because those electrons, they're not just floating into the matrix. Those electrons, even if they're generated in the matrix, they're not really directly getting to one of these complexes. So how do our electrons, or from our sugar, from our fat, from whatever the heck we want, how are they actually going to enter into this pathway? Yeah, so reduction reactions, but how are they going to be carried to the pathway? NADH is one way, exactly. So NADH, where does that come in? Complex 1. So complex 1, that's one of those ones that actually pumps out protons. So if we get the electrons in that complex 1, we pump out protons, voila, we're helping build that gradient that's going to power the ATP synthesis. What else can we use to bring in electrons? What? So shuttles, but those shuttles they were taking things in the form of other molecules, yeah. So that was one way we could get electrons in. And so keep that in mind, and we'll get back to that in a second. But what is the other kind of key way in which key carrier that we talk about? FAD and FADH2. So which version of those has the electrons? FADH2, exactly. And FAD, what was special about FAD compared to NADH? So complex 2, that's one protein that has FAD. And what's complex 2? What's its other name? Its more official name. Succinate dehydrogenase. And where do we see that? Our good old friend, that TCA. That's our succinyl dehydrogenase set. That's our complex 2 of our electron transport chain. And so complex 2, most of this stuff is happening out in the cytoplasm, but this was happening not in the cytoplasm, but in the matrix, in the interior of the mitochondria, that inner compartment. But the complex 2, it was actually in the membrane. And why did it need to be in that membrane? So complex 1 is in the membrane, but the NADH that we generate in our citric acid cycle. So in that citric acid cycle, we were generating a bunch of NADH, right? We were generating, there we go. We were generating a bunch of NADH in our citric acid cycle, but we didn't need to have these complexes embedded in the membrane. Why do we need to have the complex 2 in that membrane? Think about what's different between FAD and NAD. Can NAD just like H move around? Yeah. So NAD is like a NADH2. These are mobile carriers. So they can't get in and out of the mitochondria, but they can move around. I mean, they're soluble. They can move around. But FAD, well, that's going to be one of our flavins. And so complex 2, our succinate dehydrogenase, that's actually going to be one of our flavoproteins. And these, they're tightly held in the proteins. So basically, this FADH can move around, but our FAD, when we talk about that, it's got to be embedded in a membrane. Or I mean, it's got to be embedded in the protein that's using it. Yes. Exactly. It's a flavoprotein. Basically, the FADH2 part is like in the protein itself is the flavin part. And like the FAD itself is the flavin, and it's bound in this protein. And so we get this flavoprotein. And the protein has to be embedded in the membrane in order to get the electrons passed off. And where are they passing off those electrons to? To Q, exactly. They're passing their electrons to Q. And that's also where the NADH is passing its electrons to. They're all trying to get to Q. And then Q can take things further. But that's not the only way we can get to Q, right? Complex 2 was just one way. We can even take out complex 2. And we don't need it. So we have this like 1, 2, 3, 4, but really we don't need complex 2. We don't even go through complex 2. The electrons from complex 1 aren't going through complex 2. They're going to Q, which takes them to complex 3. We call it like complex 2, the succinate dehydrogenase, but we can imagine that there are other ways to get electrons to Q. Other different membrane embedded flavoproteins. And so we saw an example. When did we see an example of this? Back to our idea of shuttles. What were the 2 shuttles that we talked about to get electrons into the mitochondrial matrix from that NADH that we generate in the cytoplasm? Yeah, the malate aspartate shuttle, that was one of them. So our malate aspartate shuttle, basically here, we're taking the electrons from that NADH that we generate in the cytoplasm, and there we were sneaking them in the form of malate. So basically, we are reducing oxaloacetate to get malate using malate dehydrogenase, taking that through an antiporter, and then in the matrix, we were then oxidizing malate to give us oxaloacetate, regenerating, and in the process, reducing NAD plus to give us our NADH, which now is in our matrix. Where is this NADH going to then enter the pathway? Where in the ETC? Complex 1, right. And so we're going to get those protons pumped. But we also talked about a different way we could get the electrons in. And this one wasn't going to give us as big of a benefit because we were going in not at complex 1. Instead, we were taking those electrons to Q. And we were doing that using this, the glycerol 3-phosphate shuttle. With our glycerol 3-phosphate shuttle, here we were basically going to be taking those electrons and that we generate in glycolysis in the cytoplasm. We were then transferring, or some other reaction in the cytoplasm, we're generating this NADH. We're using that NADH in order to then reduce dihydroxyacetone phosphate to glycerol 3-phosphate then that glycerol 3-phosphate in the process, basically what you're having, and then you're taking glycerol 3-phosphate and then reducing FAD to get FADH2, which gets to Q. So this glycerol 3-phosphate dehydrogenase in the membrane is going to pass the electrons onto Q, which can take them onto complex 3. Yes. So it can be any sorts of NADH in the cytoplasm. So it's commonly we talk about it in terms of glycolysis, but there are other reactions we can do in the cytoplasm that will generate NADH. And then they would have the opportunity to get into the mitochondria this way as well. Where do we see dihydroxyacetone phosphate? Yeah, okay, and what was that overall pathway that we're talking about with that? Yeah, so when we talked about glycolysis, dihydroxyacetone phosphate was this guy, or DHAP. And so normally what we would think of doing is, okay, we're going to go triose phosphate isomerase, we get this glyceraldehyde 3-phosphate, which can then travel further on in glycolysis. But we can also take this dihydroxyacetone phosphate, and now what we could do is instead we could reduce it to our glycerol 3-phosphate, wherever that slide wound up. And then we can use the electrons from the glycerol 3-phosphate to then basically reduce FAD to give us our FADH2 and get back our dihydroxyacetone phosphate, which we can keep doing this with. So in this way, we're sneaking the electrons in a different way. We're sneaking them to this FAD, and then this is passing them on to Q. And this is dealing with the problem, how are we going to metabolize or catabolize? How are we going to break down? How are we going to get the energy that we're generating from in the cytoplasm when it's not coming in the form of other like carbon-containing molecules? Okay, so that's how we dealt with those electrons. We talked about how dihydroxyacetone phosphate, we are going to see that from glycolysis, but what about this glycerol 3-phosphate? Where do we see this? Yeah, lipids. When we get that backbone, that glycerol backbone, when we break down our lipids, our like triacylglycerides, we're going to get this glycerol 3-phosphate, which then we can make into dihydroxyacetone phosphate. We could use it for this shuttle. What else could we use this for? What else could we use our dihydroxyacetone phosphate for that we generated by breaking down the lipids? Yeah, so we can take it through glycolysis and we can get the energy from it. But what else can we do with it? Is glycolysis? We talk about glycolysis as being kind of, well, I mean, I guess when we think of the term glycolysis, we talk about it in kind of this one directional thing. But what else could we do? Do we have to go and break it down further? Yeah, we could go the other direction. And what do we call that other direction? Gluconeogenesis. Exactly. We call that gluconeogenesis. So what we could do is we could say, take that dihydroxyacetone phosphate, we could go and we can make some glucose from it. And so although we say like on net we can't make sugar from fats, and we'll talk more about that in a minute, but because the breakdown of a fatty acid would give you glycerol. Would give you acetyl-CoA, which isn't in the TCA. But if the glycerol part, that actually is glucogenic. So that can go and that can make glucose. But it's not in all our tissues that we can actually make glucose, right? Where can we make glucose? Where can we do gluconeogenesis? In the liver. In the liver and where else? Kidneys, right. In our liver and kidneys. And so when we talk about kind of what's happening where and in what tissues, the liver is often going to be one of those, the more complex parts, because it has all these different options that it can go through.
So let's talk a little bit more about the different options. Let's go back to that situation where we have that glucose, and that glucose is coming into our cells. Well, first of all, it kind of got to get the message to get into cells. And this is a first point of regulation that we can regulate what we're going to do with the glucose is, well, do we even take the glucose into the cells? And what's going to help influence whether we take the glucose into the cells? What's that kind of like cellular message that gets sent out when we have a lot of glucose in our blood? Insulin, right. And so insulin, when we talked about membrane, when we talked about like biosignaling, what type of a receptors was insulin signaling through? What type of receptor was our insulin receptor? It was an RTK. It was a receptor tyrosine kinase. And it can go different. Once it kind of got activated, it could do different things. And one of the different things that it could do was it could actually go and it could cause these glucose transporters to travel to the surface of cells. And so you kind of have these glucose transporters in these little membrane bubbles inside of your cells. And then when they get this message, they're then going to go take these and fuse it into the membrane. Why do you think it's a good strategy to have things kind of in these bubbles inside of the cells? Always trying to use while to actually make a whole protein to make one of those whole receptors or those glucose importers. But what's going to happen is if they're already ready to go, it's got to get the signal. Signal comes, okay, we'll come to the membrane. And now we're going to let some glucose in. Now, what are we going to do with the glucose? What are our options if our glucose is coming into our cells? Because it's not just go to glycolysis. So say glucose is coming into a liver cell. What are some of the options that it has to it? Or any cell, actually. We've got glycolysis. What did we learn about in our last lesson? Glycogen synthesis. Yes, glycogen synthesis. We'll review that in a minute. And what was kind of like the third option that we could have? Exactly, the pentose phosphate pathway. Because if we want to do lipid synthesis, what do we start with for lipid synthesis? Yeah, well, basically, we have to get to acetyl-CoA. And then we can't get the acetyl-CoA out of the matrix, and so then we have to go out as... What do we take out of the matrix to make that? Citrate. But how are we going to get that citrate from the glucose? We got to do glycolysis first, right? So we've got to break down that glucose before we can then build it into these fats. And that goes back to what we were talking about, is kind of the benefit of using these small pieces. We need to break the bigger molecules up into these smaller pieces, and then we can use those smaller pieces in a variety of different ways. But first, we got to break it down. But the other thing we could do with glycolysis is the pentose phosphate pathway. What was the things, when would we want to do the pentose phosphate pathway? What were some of the things that it could give us? That's one thing. Yeah, that's one of the key reasons why we might want to do the pentose phosphate pathway. And when did we see NADPH be needed? Lipid synthesis, right? So if we want to make lipids, if we want to make steroids, if we want to make also DNA and RNA bases, we can go down the pentose phosphate pathway. And so we didn't have time to go into the pentose phosphate pathway, but they're basically oxidative and non-oxidative forms of it.
And if you want NADPH, you can do the oxidative form of it. But if you want to do, you just need DNA, you just need your nucleobases, then you could do the non-oxidative form and still use those electrons then to go get energy from. So there are various different things that you could do from that pentose phosphate pathway. So how are we going to decide what we're going to do when? This gets back to kind of thinking about the principles of metabolism. And so this is one of the key things that I want you to focus on, and that'll be, I want you to be thinking about, and this is going to be one of the key points for the, not points like this points, but points is in, well, yeah, that kind of points too. But for your final, that's going to be a lot of the emphasis is going to be on regulation and kind of thinking about when a cell would want to do what. And yes, so in the logic of metabolism. So we talked a bit about the chemical logic, and I'm not going to ask you to like draw out any chemical reactions or anything. Well, first of all, it'd be stupid because it's open book and open note. But second of all, like this isn't a chemistry class, so I'll save that stuff for next semester's class. So I'm going to do all sorts of chemistry. But I do want you to, we did think about some of the chemical logic and the thermodynamic logic. The ways that we can regulate metabolism. We can regulate concentrations of things. And so regulating concentrations can only get us so far though, right? So when we have steps that have a really big delta G, those are going to be steps that if we change the concentrations, that's not going to be enough to really impact things. There are actually a couple of ways that concentrations can impact things. One is directly thermodynamically. So changing the concentration so that we have that K, our KEQ is, which I can never remember the directions. But basically that we have it so that we have more products or more reactants than we would need for equilibrium or whatever, however you want to say that. Basically, with Le Chatelier, we drive things that way. But if you have a really big delta G, then it's not going to be enough to change the concentrations a little bit and then drive things the other direction. But there's another way that we can have the concentrations of things impact. And that's because we don't have to be talking about the concentration of the products or the concentration of the reactants. We can be talking about the concentrations of allosteric modulators. So what is an allosteric regulator? What does that mean? Something to be allosteric. And what's an allosteric site? Exactly. So allo other, it's binding somewhere other than the active site of that enzyme. It's not binding where the reactants are binding. It's binding somewhere else and altering the function of the enzyme. And so this is one of the key ways that we can regulate enzymes and regulate activities. And one of the great things about this is that we can be regulating enzymes based on the availability of other compounds that might be coming from other pathways or coming from further down the line of the pathway or further up the pathway. There are two kind of main ways that we can think about inhibition as being either like positive or negative inhibition. So we can have an allosteric modulator bind and increase the activity. Or we can have a modulator bind and decrease the activity. These modulators can be coming from a different pathway, or they can be coming from the same pathway. If we have something like downstream, a downstream product, and it's building up, we can use that as an allosteric modulator potentially. And what would it do? What would we want it to do if we're building up our product, basically? Building up a product from down the line, what kind of message would you want to send back up to the top? Yeah, stop it, we've got enough. And so basically that would be negative feedback. But what if we have we want to prepare down the line for what's coming? Positive.
good. So we want like feed forward activation type of thing, where basically we can have something near the top of the pathway be calling down and saying, yeah, we've got some product that's going to be coming your way soon. So you should really, really prepare for that. And so that is, I want you guys to be able to kind of recognize some examples of that. Another kind of way that we can regulate activity is by regulating the enzymes through covalent modification. What was the key kind of covalent modification that we talked about that was going to affect enzyme activity? I think I heard it. Phosphorylation, right. Phosphorylation. You can quickly turn on or off an enzyme. Quickly control what's going to happen. And so what we're, this is going to be one of the key ways in which we can regulate those steps that are far from equilibrium, those steps that are hard to go back and forward from. Yes.
Yeah, things that are like far from equilibrium and the ones that are kind of close together in concentration, those more
Exactly. And that's the kind of regulation via the like thermodynamic regulation via the concentration of our reactants and our products. So what you have more of, you go the other way. What you have less of, you go that way. But basically, that is going to only be able to take you so far in the physiological sense. Theoretically, you could reverse any reaction in a test tube if you were able to have like high enough concentrations to skew things the other direction. But we're not going to have that happening in ourselves. And so for those steps, well, we can't just tilt the scale that much, because then we have to have these enzymatic regulation. And that enzymatic regulation can be through covalent covalent regulation, covalent modification, things like phosphorylation, or it could be based on allosteric modulation. So it could be allosterically modulated, and sometimes it could be allosterically modulated by its own product. So it can still be regulated that way. When we have these steps that have this really big change in free energy, those are the steps that we often tightly regulate because it's hard to go back from. What else is special about these steps? If we think about these not so much as one-way streets, but as kind of like these bigger pathways, like how do we, if we have a pathway where we have one of those steps that's really hard to go back from, how are we going to actually go back? Talk about how we can kind of like keep it from going forward, but how can we actually get the products to turn into reactions? Different enzymes, right? So we can use different enzymes. So those are the steps. Like if we look at glycolysis and gluconeogenesis, they look pretty similar. They use 7 of the same 7 of the 10 same enzymes. But those steps that we have, those big change in free energy, those steps that we have tightly regulated, those steps where it's not enough to just twiddle with the concentrations in the middle, those are the steps that we're actually going to use different enzymes in order to reverse those steps, to go around them. And so sometimes it might be actually we have to use 2 enzymes in order to get around it, such as the last step of glycolysis, or the first step of gluconeogenesis, where I need to take pyruvate to phosphonyl pyruvate. We go through these 2 steps. When we go through these 2 steps, so we get this intermediate oxaloacetate. Where do we see oxaloacetate show up? Everywhere, yeah, the TCA cycle. Basically, oxaloacetate is how we get that acetyl-CoA into our tricarboxylic acid cycle. And, well, if we think about, using the tricarboxylic acid cycle more as not just a way to get this oxidized, to get that NADH, to get the electrons in the mitochondria that we can then pass down the chain, pump the protons, get the ATP. That's not the only thing that we can use this cycle for. We can siphon off all these different components, but in order to keep the cycle going, we're going to have have to replenish components. What do we call those replenishing reactions? Anafluorotic, right? Those are anafluorotic reactions. And we have anafluorotic reactions that are going to generate this oxaloacetate. And one of the anafluorotic reactions that we could do is that step that we call like the first step of our gluconeogenesis, going from pyruvate to oxaloacetate using pyruvate carboxylase. So we see here, like, yeah, we're using this step that's in gluconeogenesis, but we're not actually doing it for gluconeogenesis potentially. We could just be doing it because we need to replenish things that were taken out of the TCA. Okay, so going back to our idea of regulation, we talked about how we could have, we have different enzymes, like separate enzymes that are going to be doing the forward and the reverse direction for those quote unquote irreversible steps. We can also have different versions of the same like enzyme in different cells. And this is going to, what do we call those? Yeah, there's like different versions of the same thing. Isomers, yeah, and it's an enzyme, so we can call it an isozyme. So those would be like isozymes. And so we'll often see that we have different isozymes in our liver than we have in, say, our muscle, because those have really different priorities. Your liver, its job, if it sees glucose, its job is not just, okay, well, I either store it or I break it down. Its job is to kind of really control the levels of glucose in your blood and control whether you like store, whether it needs to be able to store glucose more than other tissues. Yeah, your muscle cells need to be able to store it, but your liver is kind of like the real modulator, the real regulator of your blood glucose levels, of everything like that. And so your liver has different versions of things. And your liver can do gluconeogenesis, whereas your other tissues can't. And so it's going to have different versions, often different isozymes than you have in other cells. Let's go back and let's think, though, about what's going to happen when that glucose is going to enter the cell. So we talked about how like insulin signaling could lead to the import of those glucose receptors to the cell. And in order, though, to keep the glucose inside the cell, what do we have to do? Phosphorylate it, right. And where are we going to phosphorylate it? What site? Don't forget glucose 6-phosphate. This is going to trap it inside the cells. But now we have different fates for it. So let's review. What are those three fates? Glycolysis, pentose phosphate pathway. Yeah, glycogen synthesis or glycogenesis. And so if we have a lot of more sugar than we need, yeah, let's go and let's hook it up to make some glycogen. Let's store it. So let's talk a little bit about how we know that we have enough that we need and how we actually go about storing it. So what we're starting with is that glucose 6-phosphate. In order to actually get hooked up into glycogen, we're going to change it to glucose 1-phosphate. So this is phosphogluconeogenesis. It's going to take that phosphate from the 6th position, move it to the 1 position. Then you can hook it up using glycogen synthase. If we want to break down glycogen, if we need energy, then we can use glycogen phosphorylase to break down that glycogen, give us that glucose 1-phosphate, which we can then use the mutase to give us glucose 6-phosphate, which we can then take through glycolysis. Okay, and we can take it through glycolysis in order to get energy from it, or we can take it through glycolysis and then do other things with the product, such as make fat or something like this. So all sorts of different things that we could do. But we want to be able to regulate when we make glycogen and when we break down glycogen. When would we want to make glycogen? When would we want to do glycogenesis? Yeah, we got a lot of sugar.
And when we want to do glycogenolysis, when we want to break down the glycogen. Yeah, when we have low blood sugar, when we need that glucose. Now, your muscles and your liver, say, they both have glycogen. They both store in glucose as glycogen. But they're going to have really different roles when you get the signal, when they're basically sugar is needed. Your muscle cells, they're going to want to use that sugar for energy. But your liver, it doesn't just need to use sugar for energy. It also has a job of providing sugar for the rest of the body. And so the liver, when it gets the message, it's going to break down that glucose, and it's going to then release it into the bloodstream. But when your muscles have it, then they're not going to release it. And so why can your liver release it, but your muscles don't? Why do your muscles hog it, but the liver lets it out? What does your liver have that the muscle cells don't have? They don't have the G6PAs, right? They don't have the ability to take that phosphate group off. We've trapped the glucose inside of our cells, and in our liver cells, though, we've got the phosphatase. We can actually release the glucose into the bloodstream, but our muscle cells can't. So our muscle cells, they're just going to use that sugar for its energy, but your liver cells, they're going to let some out. In both of these cases, though, they need to kind of get the message to start the glycogenolysis. And they would get the message through hormone. And so the top of the process, the actual starting that breakdown is going to be regulated based on energy needs. We said that our insulin was a signal that we had lots of blood sugar. What was kind of the opposite signal? That we have low blood sugar. Glucagon, right? So glucagon is signaling there's not enough blood sugar. What do we need to respond to glucagon in order to rectify that situation, to get there more blood sugar? What type of cells do we need to respond? The alpha cells are going to be sending out the glucagon from the pancreas. But what needs to respond? The liver. The liver needs to be able to respond. Do the muscles need to be able to respond? No, they can't release glucose into the blood. So it's pointless to have them respond. But our liver needs to be able to respond. We also don't need to be able to get the signal that we need to break down some sugar because we really need energy. And so we see with glucagon and epinephrine or our adrenaline, they both can have an effect on stimulating glycogenolysis. So basically stimulating that glycogen breakdown. So if you have low blood sugar, or if you just those cells themselves need energy, then that can both stimulate the breakdown of glycogen to give you your glucose. Once you've got the glucose broken down, well, it's broken down into that glucose 1-phosphate that can again get mutated, or mutated sounds weird, but it can be changed into glucose 6-phosphate, which can then go and it can go down any of those different pathways that you want. Okay, so insulin and epinephrine are kind of going to have opposite effects, and they do so by regulating the activity of that phosphorylase and of the synthase. So would we want insulin to activate the synthase or activate the phosphorylase? We want insulin to activate the synthase, yes, because we want it to store the glucose because we've got a lot of it. We also don't want to be breaking it down at the same time that we're making it. So we want insulin to inactivate the phosphorylase. But if we have glucagon, then or we have epinephrine, what are we going to want to do then?
Yeah, we want to do the opposite. We want to activate the phosphorylase and inhibit the synthase. And so these enzymes basically do this by regulating the phosphorylase and the synthase via kind of reciprocal phosphorylation. Yes. Glucagon and epinephrine. They're both acting through G protein coupled receptors, which is one of the great things. Because with those coupled receptors, remember that the signal was getting to that membrane-bound protein. But then that membrane-bound protein was then activating things that were bound to the inside. So they would have like different G protein coupled receptors could both be attached to the same G proteins and do the same sorts of things inside of the cells. And in this case, they're both going to be activating this they're going to be activating adenylyl cyclase, our good old friend that made cyclic AMP. That was then going to activate pKa, which would activate phosphorylase B kinase, which would activate glycogen phosphorylase, which would then break down glycogen. Because they're basically both converging on the same G protein, then we can get this signal from either of these receptors. And that's one of our great examples of how we can kind of use these different receptors and these mix and match kind of parts to get the same end result from different starting signals. In the case of our insulin, well, here it's going to inactivate glycogen phosphorylase by basically activating the phosphatase that will then remove the phosphates that were activating it, and will also activate glycogen synthase. And so basically that is how you would control whether you're going down the whether you want to break down glycogen. Let's talk a minute about, though, say we want to go back to our kind of conventional idea of glycolysis and gluconeogenesis. You can imagine how we can have things be entering the pathway at all these different places. So we talked about how we can have dihydroxyacetone phosphates coming from our glycerol-free phosphates that we got from, say, breaking down lipids. We could also bring in other sugars. So we didn't really talk about it much, but we could take fructose. We could take fructose and actually enter it through this, like at this, break it up here basically, and bring it in. We can bring other sugars in. Then we can type these different components. Say we talked about how we can bring oxaloacetate in the pathway that way. So basically at these different points along the pathway, we have to make a decision. Do we want to go in our liver, we have to make the decision, do we want to do gluconeogenesis or do we want to do glycolysis? And in our other issues too, we can go different directions with things. We just don't get that same overall process of gluconeogenesis because, well, we can't like let the glucose out of the cells. We can't reverse that last step. But we want to be controlling those key steps along the way. And so you should review the different regulatory mechanisms that we see at play along these different steps. Makes sense. Like if you need energy, then you want to stimulate the breaking down. And the signals that you need energy are like you have high ADP or AMP, and you have low ATP. If you have the opposite, well then go to the opposite direction. And we also see like negative feedback inhibition from things like citrate. So if we're breaking down a lot of fats, building up a lot of citrate, basically feedback inhibition. Okay, let's say a couple quick things about fatty acids. So remember that our fatty acids, this was one of the ways we could kind of converge upon that tricarboxylic acid cycle. So if we want to make fats, we take out citrates, and if we want to break down fats, we break them down into acetyl-CoA that can then enter this pathway. Why did we take citrate out to use that instead of trying to make fatty acids from acetyl-CoA? Yeah, citrate can cross the membrane. So our lipid synthesis is going to be happening in the cytoplasm, but the breakdown is happening in the mitochondria. Speaking of that breakdown, so remember that breakdown is happening in beta oxidation in the mitochondria, and we're kind of removing these two carbon pieces at a time, these acetyl-CoAs. So I mean, like 2 carbons attached to a CoA, which is just kind of like the handle that we use. And similarly, though, when we're building up fatty acids, we're doing it from acetyl-CoA pieces that we activate through this acetyl-CoA carboxylase to give us that 3-carbon malonyl-CoA, and then we get rid of that CO2. Then we build it up using these acetyl-CoA pieces. So when you break down fatty acids, the end result is this acetyl-CoA. This last step is going to be this thiolase, this beta-keto-acyl-CoA thiolase. It's going to come in and it's going to basically break the acetyl-CoA piece off. What's going to happen if you have a lot of this acetyl-CoA build up is that this last step can actually reverse itself. If you stick 2 acetyl-CoAs together, what you get is acetoacetyl-CoA. Acetoacetyl-CoA, well now HMG-CoA synthase can transform that into beta-hydroxy beta blah blah blah. Basically, this molecule that can then get further modified to give us acetoacetate, and acetoacetate, then this beta-hydroxybutyrate dehydrogenase can transform that into beta-hydroxybutyrate. So reduce this from a ketone to an alcohol for a more stable form, this beta-hydroxybutyrate that can then transport through the bloodstream to get to tissues that need energy. And so these are what we call ketone bodies. And this is one of the ways where your liver can get energy to other tissues once it runs out of its glycogen stores, and it needs to start breaking down the fat and getting energy to other cells. Because cells like your brain, they can't use lipids directly, but they can use these ketone bodies. What happens though is that if you have too much of this build up, because say the acetyl-CoA, it can't be, your citric acid cycle is clogged up. You're generating too much of this acetyl-CoA, it can't enter the pathway, it builds up, you get the ketone body formation, you get these ketone bodies then building up in the bloodstream because your other cells don't have enough oxaloacetate to take it in. And therefore, these ketone bodies are building up. These ketone bodies have these carboxylic acids, so that the blood itself can acidify. This is ketoacidosis, and it's a key problem in people with uncontrolled diabetes. Okay, and then remember that amino acids can also enter and leave the tricarboxylic acid cycle, and they could also, some of them can form those ketone bodies. And the breakdown of amino acids happens through transamination reactions. Take off that amino group, and that leaves you with an alpha keto acid, some of which are directly in the tricarboxylic acid cycle, some of which can get to that point where they can enter the tricarboxylic acid cycle. Other than that, for them get to you to one of the ketone bodies. So you can get energy from breaking down amino acids, and you can use amino acids to get components into the cycle.